This video is a little intro to Lab 5.1. 5.1, we're looking at different growth conditions and how E. coli or Staph aureus will interact at those conditions. So the first part in this is determining the optimal temperature for growth. So week 1 activity or day 1 activity would be to inoculate your test tubes. So the first thing you would do is label your test tubes with a different temperature. So 5 degrees up to 60 degrees. Here I have my E. coli tube. We want to be aseptic, so we're going to flame sterilize our loop with the Bunsen burner. Start at the tip of that flame and get that red hot. So all tubes would be inoculated like this. Let that loop cool for a few seconds. Take the cap off of that E. coli tube with the pinky finger of that hand that you're holding with the loop. Pass the tube through the flame 2 or 3 times very quickly. Dip your loop down into the tube to get the E. coli. Reflame the tube. Recap that. Set that tube down. And then pick up the tube that you're going to inoculate. Take the cap off of that. Flame that tube. Dip the E. coli in. Reflame the tube. Recap. And then flame sterilize that loop one last time. Get the entire thing red hot so it should be ensure it's sterilized. So the first activity is inoculating those 6 Triclosan broth tubes. The only difference is the incubation time, or temperature, sorry. The second part of this, you would need 2 more Triclosan broth tubes. And we're going to look at the growth and presence and absence of oxygen. So we would simply inoculate 2 more tubes. One we would label with O2 and the other without O2. And aseptically inoculate those. The last part is determining the optimal salt concentration for E. coli. Here you'll have tubes from 0 to 5% salt, and we're actually going to look at E. coli and staph for this. So we'll have something to compare to. See how salt tolerant E. coli is compared to Staph aureus, which tends to be more salt tolerant. To actually measure the growth, we're going to use a spectrophotometer. So in the next part of this video, we'll have the results, and what you'll do is follow the lab report as I am using the spectrophotometer and recording the data for you to fill out the data sheets for this Lab 5.1 and then complete the lab report and then complete the e-learning quiz associated with this lab. In this video we're going to do some analysis of Lab 5. We're going to start with the temperature. This is our spectrophotometer. So the spectrophotometer has a light that will pass through the sample and basically record the absorbance, how much light is absorbed by the sample, which gives us an idea about the cell density. So the higher the absorbance, the more growth that we have. So what we'll do is I'm going to measure each one of these samples, and you will record the absorbance in the Lab 5.1 report. So we're going to start by taking a blank tube to make the machine think that the broth, the color of that broth, is basically clear. Make sure there's no fingerprints on this. I'm going to put it in the machine, and I'm going to hit the 0 ABS button, and that will make the machine think that that is clear. The glare, so that's reading 0 absorbance. So now what is going to be red is the actual cell density. So I'm going to save this tube just in case. start with the 5 degree Celsius tube. And as you can see, just by looking at that at 5 degrees, it's very clear, indicating at 5 degrees E. coli did not grow. But we will get a measurement anyway. And it should be right around 0. And it's at 0.012, which is no growth. So please record that data point at the 5 degree absorbance. I'll take that out. Give this a quick rinse. And go to the 25 degree tube. Now the 25 degree tube, there is some turbidity there, indicating that there is growth. Pour that into my cuvette. Put that in there, and it's reading 0.646. .30 Celsius, also turbid. I'm resuspending that by giving it a gentle little swirl there. Put that in the machine. 0.753. Again, that was 30 degrees Celsius. 37 degrees Celsius, again, lots of turbidity. .064. And I'm just taking the first data that I see. Those bacteria are settling, they're moving, floating around, settling, so I'm just taking the first data point, which is good for our application here. 45 degrees Celsius. .104. And then at 60 degrees, it's pretty hot. That tube is clear again, indicating no growth. It will have a slight absorbance because when we do incubate this media at 60 degrees Celsius, it kind of discolors a little bit. So it's reading 0.015. Which is no growth. So E. coli does not like the cold, it does not like the high temperature. So you use this data to plot that absorbance on that graph for number 2. So have a nice graph that you generate. Now, turning the page on that lab report, we're going to look at E. coli absorbance with oxygen and without. So with oxygen, E. coli grew, no problem. We're going to see how well it grew. 37 degrees Celsius with oxygen. .120. And without oxygen, E. coli grew just fine. .930. So not quite as well without oxygen, but it still did grow. But you can see it grew a little bit better with oxygen. So for number 6 in the lab report, you would classify that organism based on oxygen requirements. Obligate anaerobe, obligate aerobe, aerotolerant, facultative anaerobe. There is one answer. Now we're going to look at the optimal salt concentration for growth for E. coli. See how well E. coli tolerates that salt. And we're also going to look at Staph aureus as a comparison. So here's 0% salt. E. coli has got nice growth in that tube. This is, we're all incubated at 37 degrees Celsius. The salt concentration is what is different between these two. So for number 7, you can begin recording at 0%. The absorbance was 1.137. Next is 0.5%. .568. .1% salt. Make sure I have this right. .845. And we tend to get a little bit of jumping around with some of the data here for a while. Salt. .481. .3% salt. .353. Starting to see a downward trend in the growth. .4%. .247. And the last one here, 5%. .071. So you can see there's a nice downward trend in the E. coli's growth. As the salt concentration increases, E. coli's growth is decreasing. Use that data to plot on number 8, and you will classify E. coli as either a halophile, facultative halophile, or non-halophilic in number 10. Now we're considering a halophile something that obligate halophile requires salt for growth, and high salt concentration would be above 5% and above. Now we're going to look at Staph aureus as a comparison organism here. So you can record Staph aureus under the transmission section for number 7. Just kind of an added bonus here for you. This is Staph aureus at 0% salt, 1.240. Staph aureus at 0.5% salt, 1.005. %, salt, 0.794, % salt, 0.639, or sorry, 649. Staph aureus at 3% salt, 0.418. Staph aureus at 4% salt, 0.296. And 5% at 0.397. So you can see just from this, we don't have all the way up to 7% salt. Our experiment says we do, or should, but Staph aureus is still holding pretty strong with its growth at 5%. One of the media that we use, mannitol salt agar, has 7.5% salt. It grows no problem at that. So Staph aureus is more considered a facultative halophile. So this concludes the analysis portion using the spectrophotometer for lab 5.1. Complete that lab report, and there will be a short e-learning quiz for you to analyze.